The topic of the series is polygenic adaptation, and we are starting today with the first of the series that will have 4 episodes, which are planned for until the 25th of November. And I would like to first give floor to Antoine Kremer, who is a professor and emeritus senior researcher from INRAE, and also the former coordinator of the EVOLTREE network, who will say a few words about EVOLTREE and also introduce our excellent speaker today. So Antoine, over to you. Antoine, you need to unmute your mic.
Right, sorry for that. Hello, okay, everybody. Well, as the sessions are open to a larger community than EVOLTREE members, I will first say a few words about EVOLTREE, and then introduce our speaker today, Jonathan Pritchard. Now, EVOLTREE is a European network of labs with strong interest in genetics and evolutionary research in forest ecosystem, with trees, with main focus on trees and associated species. In total, we are about 28 labs from 19 different countries, which represent a few hundreds of scientists across European countries. EVOLTREE is now 15 years old. It was first financially supported by the European Union, and it is now self-supporting thanks to voluntary contributions, financial contribution by partner labs, or also by in-kind contribution of the different partners. It is currently hosted by EFI, which is the European Forestry Institute. So what are our objectives? Coordinate and share research efforts and resources at the continental scale. What are our activities? We maintain and upgrade common infrastructures like monitoring sites in forest and electronic resources like interoperable databases. We organize training sessions in the form of workshops and summer schools. We provide financial support for small-scale research projects. And we maintain, of course, networking activities and have disseminate material, different information material between the different partners. So far for EVOLTREE. Now today, we have the privilege to welcome Dr. Jonathan Pritchard as our first speaker. We feel very honored to have him here as speakers for our first webinar. Jonathan got his PhD at Stanford with Mark Feldman in theoretical human genetics and is now back in Stanford since 2013 as professor in the Department of Biology and Genetics. He has developed novel statistical and computational methods that provided new insights in evolutionary trajectories of human populations, but not only human populations. We all know him since he published a model-based clustering approach for inferring population structure called Structure, published in Genomics in 2000 or 2001, I don't recall exactly. Another major achievement intensively used in our community of forest geneticists is TreeMix that allows to retrace population shifts and mixtures along their evolutionary trajectory. And as I understood from his biography, the development of these tools like Structure were actually motivated by the concerns of population structure in gene association studies that Jonathan was conducting at the beginning of his career. To some extent, his interests today are a follow-up of his earlier focus on genetic architecture of complex threads. But beyond the more traditional descriptive and correlative approaches as GWAS, he proposes a more conceptual and functional approach linking regulatory processes and genes to complex threads. And this is the topic of his talk today. Needless to say that the research that Jonathan is conducting raises strong interest in our community. Trees share some peculiarities with humans, at least at the population genetic level, large population sizes, extensive gene flow, longevity, etc. And our main focus is also on complex, very integrative threads that have polygenic architecture and are strongly related to fitness. Jonathan, the microphone is yours now.
Great. Antoine, thank you so much for that kind introduction. And everybody, it's really a great pleasure to have the opportunity to talk to you today. Okay, I've gone into screen share. Okay, can you see the screen?
Yes, we can.
Okay, can you see the full slide? That's
yes.
Okay.
Yes.
Okay,
perfect.
great. All right. Yeah, excellent. Let me just minimize the. Okay, great. Yeah. Okay. So today I'm going to be telling you about work that we've been doing to try to understand the architecture of complex traits in humans. And, you know, I think that there's some advantages in thinking about humans as a model organism. We have an enormous amount of phenotyping and genotyping data. A lot of biology, of course, has been done in humans. So I'm going to tell you about some vignettes of things that we've been thinking about in the human context. And I hope that those vignettes will be of interest to you in thinking about the genetics of complex traits in trees. And when we get to the discussion part of this, I'd love to hear more about your thoughts on that. So if we go right back to the beginning of genetics and with the rediscovery of Mendel's work at the beginning of the 20th century, I think it's fair to say there's been this tension between Mendelian and biometric views of inheritance. So, of course, the biometric view was that phenotypic variation in populations seems to be largely quantitative, largely continuous, and this kind of work goes back at least to Francis Galton, and then with R.A. Fisher's 1918 paper, which gave rise to the infinitesimal model, this sort of led into the birth of quantitative genetics. And of course, we all know that quantitative genetics has been enormously powerful as a tool for plant and animal breeding, for understanding the genotype-phenotype relationship and between relatives and so on. And yet, I think it's fair to say that most of our intuition in genetics, at least how we think about the relationship between a mutation and a phenotype, comes from studies of simple and single gene traits, more in a Mendelian paradigm. So of course, one of Mendel's points of great fortune was that he happened to pick traits that were simple traits, what of course we would now refer to as Mendelian. I think it's fair to say that if he had picked many other phenotypes that are more continuous and more polygenic in nature, he wouldn't have been able to achieve the progress that he did. And the same has been true in my field, namely human genetics, that a lot of how we think about the molecular underpinnings of traits comes originally from dissecting the genetics of Mendelian disorders, starting in the 1980s and 1990s, when a large number of Mendelian disorders such as cystic fibrosis were mapped. However, of course, as we know, most phenotypic variation is genetically complex, and that's certainly true for humans. I think that's true for most organisms. We have relatively continuous distributions of phenotypic variation and enormous numbers of variants that contribute to any given trait. And so in the last decade or 15 years or so, with the advent of genome-wide association studies, it's become clear that the infinitesimal models was perhaps closer to the truth than people might have actually expected. We certainly for humans, as I will discuss, we have at least tens of thousands, perhaps even 100,000 variants that can affect any given trait. These are spread across most of the genome. And so I think this raises a conceptual question about why is it that so much of the genome matters, and how do we think about the mapping from genotype on the one hand to the phenotype on the other hand, if most of the genome actually matters. So I'm going to start by giving you 3 different vignettes from human genetics that talk about the relationship between genotype and phenotype. So, and then I'll talk a little bit more about conceptual and theoretical models that we've been working on. So the first of these is for schizophrenia. And the latest meta-analysis of schizophrenia, I think it consisted of about 200,000 individuals. It was published or posted on medRxiv earlier this year. They identified 270 genome-wide significant loci. So, of course, this is an enormous accomplishment. And yet, one of the striking things of this GWAS, as well as many other GWASs that have been done for complex traits, is that the lead SNPs only contribute a small fraction of the expected heritability for these traits. So working off an earlier freeze of the data, Bogdan Pasanyuk's lab estimated that the significant loci explain about 10% of the heritability that we expect should be there. So that seems a bit of a gap. So a dozen years ago or so, this was referred to as the mystery of the missing heritability. But now this has been largely resolved by work from Peter Vischer and many others that shows that actually there's an enormous number of additional variants that are down here in the bottom of this Manhattan plot. They don't reach genome-wide significance, but together they actually explain about 80% of the heritability. So, so we think that most of the genetic variance underlying schizophrenia is due to an enormous number of variants that are, probably acting more or less additively, and there doesn't really seem to be a major mystery of missing heritability anymore. But what I will argue is that we have a different mystery, which is why does so much of the genome actually matter for a trait like this? The last point I want to make on this is that most of the hits are very difficult to interpret. And this is very typical for common disease GWAS in humans. So in the previous published meta-analysis of schizophrenia, there are actually no gene ontology categories that were genome-wide significant, although there was sort of a hint of something going on in synaptic genes. Now in the 2020 freeze of this data, there is actually significant enrichment in synapse expressed genes, but this is a very large gene set, and it doesn't, I think in its own right, doesn't really give us an enormous amount of insight into the biology of schizophrenia, that particular geo enrichment. But anyway, I think that the GWAS of schizophrenia, in many regards, have been an enormous success. They've been able to identify an enormous number of loci that contribute to schizophrenia, as well as, in some sense, explaining a lot of the heritability. And yet, it's been shown that it's also difficult to understand the biology of what's going on. The second point that I want to make on this is that actually most of the genome contributes to schizophrenia heritability. And this is a very common sort of pattern that we see for many traits. So here, each of the bars represents one of the human autosomes, and they're arrayed along the x-axis according to chromosome size measured by numbers of SNPs. On the y-axis by the percent heritability is estimated to come from each of the chromosomes. And you can see that you can actually predict the heritability very accurately simply from chromosome size. And this is what you might imagine from a model where you have an enormous number of loci that are spread more or less uniformly across the genome that are contributing to heritability. This isn't the case for every trait. So for example, for rheumatoid arthritis, there is one big outlier locus on chromosome 6, which is here. That's the major histocompatibility locus, which is a major locus that has an effect on autoimmune conditions. But if you set chromosome 6 to the side, then you can see that actually there is a polygenic background for rheumatoid arthritis that looks in some ways similar to what we have for schizophrenia. So for traits like schizophrenia, as well as height, which is a trait that my lab studied quite a bit, as well as many other labs, of course, it's been estimated that at least 10,000, perhaps on the order of 100,000 variants likely affect each of these traits. And as I've suggested, these are spread very broadly across the genome. So, as I said before, this really raises this question: how does genetic variation map to phenotypic variation if so much of the genome matters? So I'm next going to explore the architecture of two traits that we think of as kind of model traits, namely urate and testosterone, where we actually know a lot more about the underlying biology of these traits in advance. And I'm going to use these to try to tease out a little bit about what is the architecture of the biggest hits, and as well as the polygenic background. Next, I'm going to propose a conceptual model for how we might understand complex traits. And then I'm going to talk about the role of natural selection in flattening out the largest effects for complex traits, which we think is contributing to the high polygenicity. And at the end, I'll talk a little bit about some future directions. So the second example after schizophrenia is one that we've been developing in the lab, which is urate levels. And so urate is a small molecule that arises from as a byproduct of purine synthesis. The levels of urate are tightly regulated. If you have too much urate in your bloodstream, it crystallizes into these sharp needles in the joints, which lead to gout. And we became interested in urate because it's a phenotype that was collected in an enormous data set by the UK Biobank. So the UK Biobank is a study of British individuals. They measured urate as well as about 30 other metabolites in more than 400,000 individuals. And this is a project that was initially led by, or initially started by a graduate student in my lab, NASA's Sinead Armstrong, working with Manny Rivas. And then more recently, NASA, as well as a postdoc in my lab, Sahin, have taken this up to really try to understand a lot about the biology of these individual traits. And so this work's described in a pair of articles on bioRxiv. So if you look for NASA Sinead Armstrong, you can find both of those. So one reason why we got interested in urate is that, unlike schizophrenia or most other disease traits, we actually know a lot in advance about the processes that might be expected to regulate urate. So first of all, urate comes as a metabolic byproduct out of this biosynthetic pathway. So everywhere where you see an arrow that's a transition between two molecules on here, we actually know what the genes are that catalyze those steps. So all of those are natural candidates for controlling urate levels. Secondly, we know a lot about the regulation of urate in the bloodstream. So urate is produced as a byproduct in all tissues, and then it gets pumped out into the bloodstream. And the levels of urate are heavily controlled by the kidneys. So there are these transporter genes that are marked here in gray that are responsible for pumping excess urate out of the bloodstream into the kidneys. And when it's in the kidneys, it can either be excreted through urine or it can be re-uptaken into the blood. And so again, this is a case where we actually know all of these transporter genes from prior physiology work. And so again, these might be expected as natural candidates for driving urate levels. Okay, so now we can go ahead and do a GWAS for urate in the UK Biobank. And here are the hits. Remarkably, we identified more than 200 independent genome-wide significance signals. A second point that was really striking to us immediately was that actually many of the lead hits are directly associated with these urate solute carrier genes. So all of the genes that are marked in green here are urate solute carriers in the kidneys. And so you can see actually that most of the urate carriers that we had annotated from the prior literature actually show up as genome-wide, as being among the biggest genome-wide significant hits here. We also see a whole suite of genes that are transcription factors that are involved in kidney and liver development that show up as being among the larger hits. And we also in red have a couple of large hits that are genes that are involved in many biomarkers, but where we don't really understand what the mechanism is. But we might speculate that these are involved in some way in kidney development. So if we take those results and put them back onto the picture that I showed you before, we see that actually remarkably, 8 out of 10 of these solute carrier genes that we'd annotated in advance have genome-wide significant signals. So at least by the standards of human genetics, that's really a remarkable enrichment. It's about 37-fold enrichment, and as you might expect, it's massively significant. We can also go back to the biosynthetic pathway that I told you about before. And here we again see enrichment, although it's actually much less remarkable than for the solute carriers. So every gene here that's marked in red has a genome-wide significant hit. And the numbers in gray indicate additional genes that we had annotated as being on this pathway that do not have genome-wide significant hits. So here we get 16 out of 88 of the genes have a genome-wide significant hit. That's about a 2-fold enrichment. That's significant, but it's it's perhaps not as compelling as we had for the solute carriers. So right off the bat, this kind of suggests to us that regulation in the kidneys is really the most important factor that's driving urate levels, and not really the synthesis end of it. And I'll come back to this in a moment, but despite these big hits, actually most of the heritability is driven by a large number of SNPs that are in kidney regulatory chromatin. Okay, so the next example that I'm going to show you is for testosterone. And this is another small molecule that's circulating in the bloodstream, also collected by UK Biobank. And again, we got interested in testosterone because it's a molecule that we know a lot about from prior biology. So as you will know, testosterone has anabolic roles in building muscles in both sexes, as well as secondary sexual characteristics, particularly in males. The levels of testosterone are dramatically different in males versus females. It's about a 10-fold difference. So if you see here, the x-axis is on a log scale. So there's about a 10-fold difference in the mean levels of testosterone between the sexes. Again, for testosterone, as for urate, we know in pretty much complete detail that the biosynthetic pathway of testosterone, it comes out of this cholesterol biosynthesis pathway. Here it is. So there's a bunch of molecules in this that whose names you recognize: cortisol, estradiol, aldosterone, and so on. So the testosterone is an androgen, so it's one of a whole set of molecules that are involved in males' sexual secondary characteristics. So, so we decided to, based on some preliminary analysis, we decided to do the GWAS for male and female testosterone separately. So here's what the results look like for females. So of course, here the sample size now drops down by a factor of 2. So we have about 150,000 individuals in this analysis. What we find for females is that actually most of the lead hits are involved in the biosynthetic pathway. So all of these hits that are in green are involved in the biosynthesis. We also have a couple of hits that are involved in gonadal development in females. And so this is starting to give us a picture that's somewhat contrasting to what we saw for urate, where we found that biosynthesis is actually not that important. We also find that the patterns for male testosterone are almost completely different. We do get some hits that are involved in the biosynthetic pathway. These hits in green, but actually the biggest hits are involved in homeostasis of testosterone in the bloodstream. And I'm going to talk about this more. SHBG is a binding globulin protein that binds onto testosterone when it's circulating in the bloodstream. So this is the biggest hit in males. It's not a hit in females at all, for reasons I'll describe in a minute. So one thing that really struck us when we were looking at this is that male and female testosterone are essentially unrelated traits. So if we take the lead variants that are genome-wide significant in either males or females, and then we look at their effect size in the two sexes jointly, we see there's essentially no correlation at all. When we look genome-wide using LD score regression across all variants, again, we see the genetic correlation is essentially zero. And so this is maybe a little bit of an aside, but I wanted to talk about this a bit because it's something that we found really striking. So first of all, what we find is that the steroid synthesis part of these processes is important for both sexes, but it's actually different parts of the pathway that are most important. So this plot, I'm going back to the steroid synthesis pathway that I showed you before, but now we're adding in the gene names. So all of the gene names that are in purple are close to genome-wide significant hit in males. All of the hits that are in orange are genome-wide significant females. And then there's a couple that are genome-wide significant in both. These are marked in black, although even for those, it actually winds up being different SNPs for males and females. So what you can see here is that it's actually a different part of the pathway that has most of the female signal, while most of the male signal is in the androgen part of the pathway. We think that part of what's going on actually is that a lot of the female testosterone is produced in the adrenal gland, which is responsible for producing cortisol and aldosterone. And then testosterone is sort of a byproduct of that. Whereas, of course, most of the male testosterone is produced in the testis, which really emphasizes this part of the pathway. And then the last point I want to make on this is that we actually see that there are really key differences in the regulation of testosterone as well as the main tissues of production. So male testosterone is heavily regulated through something called the HPG axis, which is the hypothalamus and pituitary in the brain. These signal, these send a molecule called luteinizing hormone that stimulates the testis to produce testosterone. This circulates in the bloodstream. It's about half of the testosterone in the bloodstream is bound by this binding globulin called SHBG. SHBG actually makes testosterone bioinactive. The active part of the active fraction of the testosterone then in turn has this negative feedback loop, which is blocking LH production. So when you increase SHBG levels, what that does is it actually reduces the negative feedback loop, so you actually get more total testosterone produced. It turns out that in females, we don't really have this negative feedback loop to any extent because the testosterone levels in females are low enough that they don't actually trigger this. So the SHBG, so there's no real regulation by SHBG in females. We can actually blow up this upper part of the signaling pathway. We know what many of the genes are in this in the hypothalamus and pituitary part of the pathway, and we see that actually more than half of these have genome-wide significant hits. So all of the genes that are marked in purple do have a genome-wide significant hit. The ones in gray do not. And I'll note that all of these genome-wide significant hits are from males. Actually, none of those are significant in females. And then, of course, as I said before, the single biggest hit in the genome for males is at the SHBG locus, as well as several other genes that are regulating SHBG. So I've gone through this in large part to illustrate that unlike for something like schizophrenia, we find that for these molecular traits, the lead variants really highlight these core pathways to an extent that is extremely rare to see for disease traits. And so I think this raises a question, or you may be wondering, if we're seeing such strong enrichment in these core genes and pathways, are these traits actually polygenic in the same way that I discussed for height or schizophrenia and so on? And in short, the answer is yes. They are also very highly polygenic. So we've done an analysis where we chopped the genome up into windows of about a megabase apiece. This is sort of a bit larger than the extent of linkage disequilibrium in humans, so we can get approximately independent windows. And then we estimated the amount of SNP heritability that comes from each of these windows. And these plots show a plot of the cumulative heritability along the genome from chromosome 1 to chromosome 22 in males and females. And what you can see here, first of all, is that actually most of the genome is contributing to testosterone heritability, like fairly uniformly. Like there's a couple of, there's a few regions of the genome that contribute disproportionately to heritability. So for example, in purple here is the region that contains the SHB gene in males. So this is the largest single contributor. But overall, you get a sense that actually the heritability for testosterone is really driven by genome-wide effects. And we can calculate how much of the heritability is coming from these core pathways, and it's about 10% in both males and in females, although the actual pathways that are most important are different. A second point that I want to make is that for both traits, actually most of the heritability is coming from a large number of small regulatory effects. So one thing we can do is to look at active chromatin that's estimated using histone marks or a TAC-seq to identify open chromatin. And we find that when we look at active chromatin in kidney, if we look at chromatin that's active in kidney cells, we find there's about a 37-fold enrichment of signal in SNPs in those regions relative to the genome-wide background. And in fact, that can actually explain most of the signal. So if we take the kidney active regions out of the analysis and then repeat that for other tissues, we see that there's very little signal coming from other tissues. Some small contributions to be sure, but most of what's going on is really just driven by an enormous number of SNPs in kidney active regulatory regions. And we've done some more analysis that I won't get into, but we estimate that about, depending on the trait, somewhere between 4,000 and 12,000 variants are affecting each of the traits we've looked at, so urate and testosterone, as well as one that I haven't talked about, which is IGF-1. Using the same method, we estimate actually more like 80,000 variants may be affecting height. So just to recap this first half of the talk, what I've shown you is that for a variety of different traits and diseases, we find that the heritability is spread very widely across the genome. We find that genes with trait-relevant functions generally are contributing just a small fraction of the total disease risk. The biggest hits do show enrichment in relevant gene sets. There are genes that are particularly important, but actually most of the heritability is coming from an enormous number of variants spread widely across the genome, and they tend to be an active chromatin. And this really implies that most of the effects of heritability are being mediated through gene regulation. And this is something I will come back to as an important point in the next few slides. So I think this raises a question. How should we think about the molecular links from genetic variation to complex traits in light of these kinds of observations? So in a series of papers, we've been developing a conceptual model that we refer to as the Omnigenic model, or sometimes the core gene model. And I'll tell you a little bit about that. We have a couple of longer papers that we published in the last few years, particularly this Chuanyao Liu and Yang Li paper, where you can go to see more of the modeling and discussion about how we think about this. So in our way of thinking, we think it's helpful to have sort of a conceptual partition between a set of genes that are relatively more important, in particular genes that have direct effects on phenotypes. These are what we refer to as core genes, as well as peripheral genes, which are basically all of the other genes that are expressed in relevant cell types. So when we think about genetic variation as affecting a phenotype, it could do that in one of two ways. It could either be due to SNPs that are either inside the core genes as missense variants, for example, or they're cis-regulatory variants. And so they have relatively direct effects. So these SNPs affect regulation of core genes, and then those core genes have a direct causal arrow to the phenotype. But also, we can have SNPs that are elsewhere in the genome. They are perhaps cis-regulators of genes that are not particularly related to the phenotype we're looking at. They are cis-EQTLs to these peripheral genes. They may be trans-EQTLs to the core genes, simply through effects that percolate through trans-regulatory networks. And so these SNPs can also have effects on the phenotype, but they're going to tend to be smaller because they're relatively indirect effects. But what we argue is that there's actually so many of these transacting peripheral variants that they actually drive most of the phenotypic variance. So to plot this a different way, we sort of imagine that the core genes are embedded in cellular regulatory networks. Core genes, by our definition, have direct causal arrows to the phenotype, but actually most of the heritability is coming from this huge number of peripheral variants that have individually small effects on core genes. And so just to state our hypotheses a bit more directly, what we propose is actually most of the heritability is coming indirectly through these weak trans-regulatory effects. We propose that essentially every gene that's expressed in relevant tissues is connected to these regulatory networks, and thus has the opportunity to have non-zero effects, although of course many of these may have effects that are very close to zero in practice. And then following on from hypothesis 2, what this means is essentially any SNP that's a regulatory variant in a relevant tissue can have non-zero effects on the phenotype. And our argument is that actually there's a huge number of variants that fit into this category, and that's what really drives the polygenicity of complex traits. So it's helpful now to, okay, so let me say that like in general, we don't really know enough about what are the causal variants for complex traits, as well as what are the regulatory networks, to really actually sort of flesh out this in real data in full. But what we do know a bit about is some things that are related. Like in particular, we know about the importance of cis and trans regulation for genes. So, what we imagine this model is that there's a set of SNPs that have relatively direct effects because they're in sister core genes, as well as trans variants for peripheral genes. Now, if you've ever done EQTL mapping, one thing you may be worrying about is that we know that trans-EQTLs generally have very small effect sizes compared to cis effects. However, it's become clear in the last few years that actually a lot of the variance in gene expression actually comes from trans effects. So I did a literature review last year where I looked for studies of cis and trans heritability of gene expression from a variety of different organisms and different tissues. And what we find in that literature review is that somewhere around 70% of the heritability of gene expression for typical traits is coming from about 70% of the heritability in gene expression for typical genes is coming from trans effects. And so so we can now start to put some numbers on this picture I showed you before. So we estimate that about 70% of variance is coming from trans effects. However, as I told you before, trans associations tend to be very much weaker than cis effects. And what this implies is that a typical gene must really have enormous numbers of very weak trans-regulators to square these two observations. So if we imagine that there are at least tens, probably hundreds of core genes for any given trait, and each core gene has enormous numbers of trans variants, then this model starts to explain why such a large fraction of the genome contributes to any given complex trait. And if you're interested in more in this, we have some mathematical models in this paper that we published last year. So that's one part of what we think is going on. But a second kind of question is, why are the big effects at core genes so rare? And so this is something we've been tackling theoretically using work with my postdoc, Yuval Simons, as well as my collaborator, Guy Seller, who is Yuval's PhD advisor. And they have an important paper on the genetic architecture of complex traits that they published in PLOS Biology a couple of years ago. And so their model starts from the point of view that most traits are probably under some degree of stabilizing selection. And what this means is that for most traits, there's some optimal trait value, and then you have a balance between two kinds of forces. So mutation tends to increase phenotypic variance, but natural selection tends to reduce phenotypic variance by decreasing the fitness of individuals who are in the tail of the distribution. So if you think about testosterone levels or urate levels, it's likely that having testosterone levels that are too high or too low, on average, will tend to decrease your fitness. So through the work that Yuval has done, as well as more classic work and from Landy and others, what we know is that in this kind of model, selection can, in some sense, see big effect variance, but not small effect variance. So in a sense, you can think of variance with small effects as kind of getting through the sieve of natural selection, while big effect variance tend to be purged by purifying selection. So the next point I need to make as a setup is that when you think about the contribution that a SNP makes to phenotypic variance, it depends on the following. So the contribution depends on beta squared. So here beta is the effect size of each allele on the phenotype, and then P1 minus P is or 2P1 minus P is the heterozygosity, so P is the allele frequency. So basically, in order for a SNP to contribute substantially to the phenotypic variance in the population, it needs to have both a reasonably large beta squared, as well as to be common enough that it can actually contribute a non-trivial amount. So variants that have big effect sizes but are very rare don't actually wind up contributing very much. And similarly, variants that are common but with very, very small effect sizes also don't contribute very much. So we can look at the contribution to heritability as a function of effect size under the model that Yuval and Guy have developed. So just to orient you here, the x-axis on this plot is going to be a measure of the squared effect size, and the unit's going to depend on population size. And this is on a log scale. And then again, on the y-axis, it's going to be a measure of the relative contribution to the variance. So first of all, if we look at variants with small effect sizes, the amount that they contribute to the heritability in the population is simply proportional to their squared effect size. And this is simply because small effect variants are not really visible to selection. So they're essentially drifting like neutral alleles. What happens when you get to larger effect sizes is that you actually get this plateauing effect where the large effect variants wind up being rare and contribute a relatively modest amount of variance. So in this part of the plot, you actually get an inverse relationship between the heterozygosity of the variance and the squared effect size, which means that very, very important variants with scaled effect sizes like 100 or 1,000 don't actually contribute more to the variation in the population than do variants with effect sizes that are on the order of 5 or 10. And there's also this interesting hump here, which comes from variants with relatively intermediate effect sizes. And it turns out these are variants that can sort of transit across to intermediate frequencies. And because of the nature of the stabilizing selection model, it actually, you tend to have selection against the minor allele. So if they can transit across to allele frequencies near 0.5, selection actually gets reduced in that part of the allele frequency spectrum. So we like to think of this as being sort of like a flattening effect, where the loci that are biologically most important are actually contributing less to the phenotypic variation than you might expect, while all of those variants that are, weak trans-pQTLs, peripheral loci that really contribute very little individually, those don't really pay any cost at all from their effect on the phenotype. And I'll note that this kind of point has also been made by Luke O'Connor in a paper that he published last year, where he referred to this as a flattening effect. So we think that this is probably part of why the largest effect variants don't contribute more, but I do want to make the point that there are exceptions to this. So for example, in mammals, we know that the genetic architecture of traits can vary dramatically across species. So this is a picture of a very large dog, very small dog, and it turns out that this size difference is driven in large part by a variant in a single gene, IGF2. This is from important work by Elaine Ostrander's lab about 10 years ago. What she found was that something like 90% of the variation in body size in dogs is driven by something like on the order of half a dozen genes, whereas body size in humans is one of the most polygenic traits that we know of, and it's driven by more than 100,000 variants across the genome. And so what we might imagine is that when you have very, very strong directional selection, as happens in animal breeding, this can probably override these negative pleiotropic effects, and it can sort of dramatically reshape the genetic architecture of a trait like body size. In the last few minutes, I just want to point to some work that we're doing now in my lab, which is trying to do a better job of linking peripheral regulatory variants to core genes. And so one of the real challenges in interpreting this core and peripheral gene model that I've posed to you is that in general at present we know very little about what are the trans-regulators of any given core gene. And in principle, one could measure this using trans-pQTLs, but at least in humans, the effect sizes are so small that it's extremely difficult to get to the sample sizes that we need. We probably need on the order of tens of thousands of individuals, and that's not practically feasible for most tissues, like most tissues that we can't access. At least people are not really willing to give us access to most of their tissues. So in collaboration with Alex Marston's lab at UCSF, we've been working on CRISPR-based approaches to studying regulatory networks as an alternative to trans-pQTL mapping. And I think this is a direction that's going to really allow the field to make huge progress on measuring regulatory networks. This is, of course, an area where a lot of different labs are now putting a lot of focus. So we had this idea, along with Jake Freimer, who's the postdoc leading this project, that we could do what I think of as trans EQTL or trans-protein QTL mapping in a bottle. So we do this for primary T cells. So here's basically what the idea is. So we have a gene that we're interested in, and we can put a fluorescent tag on this gene. And so we can use this fluorescent tag to measure whether that particular protein is expressed at a low level, like in this particular cell, or at a high level, like in this particular cell. We can put those cells onto a fax sorting machine, and we can sort the cells into a distribution according to whether they express this protein at a high level on the right or a low level on the left. Next, in parallel, we can couple this to a CRISPR knockout library so that for every other gene in the genome, we have a CRISPR guide that has the potential to knock that gene out. And so we set this up so that each cell gets on the order of one knockout guide. And so now we can ask whether a particular guide is associated with increasing the level of that particular target gene or decreasing it. So basically, if you find that knockout of a particular gene marked in green here tends to increase the level of the gene of interest, then you'll get enrichment for those guides in the right-hand bin. Conversely, if it for the blue gene here, if the knockout tends to decrease the level of the target gene, then you'll get a decrease over here. So what we do is we run the cells through this fax sorting machine. We sequence the guide libraries from the low bin and the high bin, and then we look for differences. So we've started this with initially a guide library of about 1,500 guides against human transcription factors. And we've done this in a particular cell type called T effector cells, which are critical in T cell mediated immunity. And this plot shows you the guide enrichments in two different donors. So donor 1 on the x-axis, donor 2 on the right axis. Every dot here shows you the value for the knockout of a particular gene. All of the genes that are marked in red are significant hits. So what you can see is that we get very, very strong agreement between the hits from donor 1 versus donor 2, which shows us that we can really replicate this study well. And Jake's actually done a lot of follow-up analysis to really verify that these hits are real hits, and also to actually start to really flesh out these networks around the genes of interest. We can do this in different cell types. So this is doing this in T effector cells on the y-axis, which is the cell type I showed you before. T effectors have kind of an activating role in immunity. Plotted against T regulator cells, regular Treg cells have basically a dampening effect. So your the strength of immune response that you have is basically reflecting a balance between the crosstalk between Treg and T effector cells. And what you can see here actually is that we have a bunch of genes that are having similar effects in both cell types. These are in red, but we also have some cell type specific effects. These are marked in blue and in green. And so this is something we interpret as showing that we actually have different trans-regulatory networks in the two different cell types. And I should mention that the Treg data are a bit more preliminary than the T effector data. So we think that this kind of approach is going to be really valuable for starting to understand the regulatory networks that drive a lot of the weaker GWAS hits. So what I've told you about today is our model for understanding why so much of the genome matters. As a conceptual framework, we propose that it's helpful to think about two classes of genes, what we think of as core genes, which have relatively direct effects on phenotypes, as well as peripheral genes. And we propose actually most of the heritability is flowing through a huge number of trans-EQTLs from peripheral genes. It's important to note that the the division between core genes and peripheral genes can be a bit blurry in some cases. We think this is still useful as a way of thinking about the structure of the data. For disease biology, in most cases, we'd be interested in identifying core genes as well as master regulators. But for other aspects like polygenic prediction, as well as perhaps polygenic adaptation, what we care about most are simply the effect sizes. So I'll talk about this in the next slide a little bit, but we may not really care very much for polygenic prediction in terms of adaptation. Adaptation may not really care very much whether it's acting on core variation or peripheral variation. So just to set up some of the things that I think are interesting about, or interesting open Popgen questions. These are some of the questions that I don't think we have really a great handle on, certainly not in humans, but perhaps not really in any species. So one kind of question is, what are the roles of selective sweeps at individual loci versus polygenic shifts in driving adaptation? I think it's becoming clear that actually the relative importance of sweeps versus polygenic adaptation may be very different in different kinds of species. Secondly, can we detect polygenic adaptation robustly? We've had some stumbles in doing this in humans. I can talk about this a little bit more in the Q&A. I think it's clear that there must be a lot of polygenic adaptation going on in humans, but it's really been a bit difficult to distinguish adaptation from artifactual signals that come from weak effective population stratification. Another question that I alluded to a little bit is, what are the settings in which we see selection driven by core genes versus by polygenic background? So the artificial selection that we see in dogs is pretty clearly driven by core genes, but probably in a lot of other settings, it's really going to be the polygenic background that is the driver. Another kind of question is, if most genes can affect any given trait, this kind of implies that there must be an enormous amount of pleiotropy across different traits. And I think a really important question that's both a theoretical question as well as an empirical question is, does this constrain the ability of a population or a species to adapt to different adapt polygenically to different pressures at the same time? And similarly, are there limits to how many different orthogonal traits we can select simultaneously? So lastly, I just want to thank the key people who have been involved in many aspects of this. So our work on the Omnigenic model came from, was started by Evan and Yang when they were in the lab. We have a theory paper, which was led by Chuan Yao, also with help from Yang. The population genetics work that I described to you was led by Yuval, and that's work that's still in progress. NASA and Sahin led the work on the small molecule GWAS. That's on bioRxiv, you can find it there. And then Jake Freimer, in collaboration with Alex Marston's lab, has been doing all of our work on CRISPR perturbations. So I'll end there and take any questions that you might have.
Thank you very much, Jonathan. That was excellent. Thank you for this talk, and we are all thrilled to have you here, and also looking forward to asking some questions and having a little bit of discussion, because as EVOLTREE, we wanted to offer this space for scientists who are interested in the topic to engage with you and to have a possibility to talk a little bit. So before we move on to questions, I just wanted to briefly explain the technicalities of this. There is a button under the list of participants where you can raise your hand. So you can either raise your hand and then I will ask you to unmute yourself and ask the question, or you also have an option of writing your question in the chat. So we just a little remark here that the session is being recorded, and we are also streaming it onto YouTube of EVOLTREE. But that doesn't mean that you have to be shy. Don't be shy. Ask all the questions that you want. And there you go. So the floor is open to the audience. And we've got Tanya, and also another little question for you to please introduce yourself and also say where you're from. So Tanya, please.
Hi, I'm Tanya Pyhäri from University of Oulu, and you mentioned that in different kind of species, the adaptation may be through sweeps or through polygenic adaptation. So what kind of traits do you think would affect which route? Like I was thinking, does genome architecture, if it's very condensed or is it very large, like in conifers, could that have an effect? Which is more likely?
You know, I think this is a fascinating question, and I don't have a good answer to it, but actually, I'd love to hear about your thoughts from a plant perspective. But thinking about vertebrates, we see patterns like, you know, for example, sticklebacks that sort of adapted to freshwater environments after the last glaciation. Those show huge numbers of hard and soft sweeps that drive, like, even sort of changes in like sort of body plan, like they have these pelvic spines that appear when they, or sorry, that disappear when they move into freshwater habitats. And these are driven by individual major effect loci. There's you know, and there's the dog examples that I showed you, and then humans have you know, very, very few examples of this kind of thing. There's a handful of relatively strong sweeps in contrast to the stickleback example. So I think in dogs, you know, most of this is driven by artificial selection, and it may be that's not really representative of what goes on in nature. The stickleback example, you know, my reading of that is it looks really fundamentally different from what we see in humans, and I think that, you know, one kind of important difference may be just sort of the you know, kind of the life history strategy of a small fish versus a long-lived mammal. You know, so you know, the fish can produce hundreds of eggs. They have, you know, very short generation time. I think you can actually sort of spread an advantageous allele under, you know, sort of very strong and effective selection, like through into a new habitat very rapidly, in a way that humans really can't. So, you know, humans, you know, if you think about like the time scale of adaptation for humans being, you know, at least, let's say, tens of generations under polygenic adaptation, you know, the range of environments that we're going to see, you know, that a population is going to see over that time scale, you know, it's going to be quite considerable, and, you know, they're going to be seeing different, you know, differences in space and geography, and, you know, our fecundity is low compared to fish. So I would. Yeah, I guess I'd love to hear what your thoughts are for plants. I mean, I think that, you know, plants have like trees can have aspects of both of this, right? So obviously very long generation time, but also the opportunity to produce huge numbers of progeny.
Santi, would you like to comment on that?
Actually, it's an independent question. I don't know if someone wants to comment on Tanya's question and the reply by Jonathan.
Yeah, but Tanya, I mean, I think this is actually a really important question that we don't have a good handle on. And actually, I mean, to begin with, I think it'd be important to have more examples of, or actually, I mean, you know, probably even just sort of somebody tabulating the literature. I don't know if this has been done carefully to understand to what extent different organisms are similar or different in their modes of adaptation, and then actually to understand why this is. To me, it seems like an underexplored question in the field.
Yeah, I was thinking that in species like conifers with the huge genomes, that they have this bigger functional space, that there is more chances for regulatory variation because the coding region is so tiny part of the genomes, which might maybe lead to more of the polygenic adaptation than to sweeps. But who knows?
I see. That's interesting. Do we know if there's, like, is there is there actually a larger fraction of the genome is regulatory in these genomes in these very large genomes, or is it? I mean, a lot of it's probably TEs and other kinds of not very less functional sequence.
I haven't seen that looked at from the conifers yet, but at least in some to some extent it seems to be so. I've seen a comparison from based on the open chromatin from maize, Arabidopsis, tomatoes, and there seems to be more of open chromatin in larger plant genomes.
That's very interesting.
Okay, Santi, please go ahead.
Yeah, I just wanted to ask about well, one of the open questions is how we deal with population stratification when we do this kind of study. So I would like to know what is your current views on that. If we will manage, do you think we will manage, and how you have any hint of how to deal with it? Like, because this is a very common problem in forestry, where actually they normally have a very wide range distribution and quite a strong population structure. So,
yeah. So I think that's a really important question, and maybe some of you may not be familiar with this story, but I can tell you a little bit about our experience in humans with this, with population structure as a problem. So the first studies of polygenic adaptation in humans focused on height, and the reason was that actually the like GWAS for height had very big sample sizes before most other studies, and then also, of course, average height varies quite a lot across different human populations, and so there was a question about whether this might be adaptive, and so there was a paper from Joel Hirschhorn's lab at Harvard, and or the Broad Institute, and they concluded that they concluded that alleles that are associated with being taller tend to be at higher frequency in Northern Europe than in Southern Europe. And based on that, they concluded that there was strong evidence for adaptive evolution driving an average difference in height across Europe. And then there's a number of other papers that did different kinds of analyses, but they all come to roughly the same conclusion. And we have one of those papers in 2016, and we argue that actually a large fraction of the genome has been affected by this. So that was all of those those analyses were building on one data set, which was a large GWAS called the GIANT study. And then starting around 2017 or 2018, we started to get access to a newer data set called UK Biobank, where the population structure is much better controlled. And it turns out that most of that signal goes away when we use the UK Biobank data. And so it seems pretty clear that there was some contamination from population structure in the GIANT analysis, and that actually misled most of the studies in this field. And so there's still some debate about whether there is or is not some signal for selection on height in Europeans. My gut feeling is that there is, but it's certainly much weaker than we thought initially. So I don't really think that there's a silver bullet to solving this problem. If you can get really clean data in a common garden kind of setting, that can make the GWAS stronger. In like one kind of data that can really help a lot is using data from relatives. So in humans, we think about like sort of SIB pair design. In plants, you have the opportunity for sort of having huge SIB pair controlled designs. So I think that having very carefully controlled experimental data is much better for this than association study data that are more opportunistic. So I do think that like the opportunistic data can work well for identifying biggest hits and core genes and things like this, but the study of polygenic adaptation is just exquisitely sensitive to population structure, because it's almost the same kind of signal, and so like you have to be much more careful with the GWAS for those kinds of questions.
Thank you. Yeah, I think that's actually a big difference, and so one of the few things we are a bit lacking for these trees, because we can't have even clones. So that for some species, at least those that have some commercial value, they are quite large common gardens, and that is a good point that you made. That in that way, I think is in one sense we are in good shape there for controlling for population structure. Like yeah, thank you.
We've got Antoine Kremer next. Antoine, can you please unmute yourself and maybe put your camera on?
My mic was...
Your mic is now working, yeah.
Okay, now that's interesting. I mean, the comments that you made just now, because we come from the plant world where we used to work on common gardens, but actually evolution is going on in natura, in situ. And what you get in common gardens and in experimental sites would not, sometimes not, and most of the times not be informative enough for the real world, for in situ conditions. So it's a challenging issue. You may have nice results in common gardens, but how far can you extend these results then to what's actually going on in terms of adaptation in natura? And then my question was, you started your talk with the infinitesimal models, the polygenic model, and I was just curious, knowing now by subdividing the genes of the polygenic model into core genes and peripheral genes, how far is the comparison working between the biometrical model and new model in terms of effects? I mean, I understand that the direct effects with core genes are likely to be additive effects, but do you also introduce interactions between genes like dominance or epistasis? And then of course there's the covariances between all those genes that inflate, I mean, the genetic variance. How do you separately estimate the covariance between the peripheral genes and the core genes, or how is this working?
Yeah, that's a great question. So in the models that we use, like everything is additive, and so for example, when we're estimating how much of the phenotypic variance is explained by SNPs, like that's a fully additive model, both in terms of dominance as well as in terms of SNP interactions, and the same is true for sort of polygenic prediction models, and so that's partly partly just a pragmatic choice that it's very difficult to estimate interaction effects. But it also reflects that, at least in humans, we find it extremely difficult to actually detect any interactions. So even among the biggest effect loci, it's extremely rare that we find significant interaction effects. And I think this is another of these features that varies enormously across species. And like humans are, in some sense, quite a young, like a diaspora out of Africa is really quite a young process. So in coalescent terms, that's like 0.1 n generations, something like this, since the diaspora out of Africa, and there really hasn't been time for sort of epistatic combinations to evolve in different populations, and perhaps that's why. I mean, people who work in people who work in mice or in flies report a lot more epistatic interactions, and perhaps in the species you work with as well, you see that. I don't know.
And then still, in addition to that, as I understood, the trans effects are translated in biomathematical terms into covariances. Okay.
Excuse me, say again, please.
The trans effects between the core genes or between the peripheral genes introduce covariances of gene effects and then contribute.
Mm-hmm.
But in a population, these covariances are likely to get erased by recombination. How stable are those covariances?
So you're referring to the covariance terms in the trans model, is that right? Yeah. So our notion is that these are not covariances in terms of LD, but covariances in terms of the gene regulatory network. Mm-hmm. So
yeah, okay.
So you might imagine like let's say two transcription factors, both are both regulating the same. Mm-hmm. Actually, let me say it differently. Like if a transcription factor is regulating two different core genes, then that induces a covariance in the expression of those two core genes.
Yes.
So, yeah. Thank you.
Thank you for this. We've got another question from Fabio Marroni.
I am Fabio Marroni from University of Udine. Thank you for the talk. And I was really fascinated by the CRISPR perturbation approach. And I was wondering if one could use such approach also to test eventually the variation in either eQTLs or regulatory network that happens after you induce a change with CRISPR. Instead of, because you basically, and I was also curious why you prefer to use a pQTL approach. It is technical or there's some reason?
Yeah, okay. So to the second point, do you mean is like why we're preferring to measure protein rather than mRNA, for example? Yeah,
yes.
Okay. So two reasons for that. One is technical, that we put a fluorescent, like we use an antibody to tag the protein. And so just from a technical point of view, like we have good antibodies for many of the genes we're interested in. So that's helpful. I mean, I think one could do that with like a fish kind of approach for mRNA as well. But anyway, we do that with protein. But actually another reason was that most of the techniques that are in this kind of space use RNA sequencing as a readout. And so we don't actually know very much about the layer of regulation that's taking place between mRNA and protein levels. And so we actually think it's quite valuable to have a readout that's focusing on proteins. And at this point, we have a few examples. We have some examples of genes where we think that the major effect is on the protein and not on the mRNA. And these are genes that are involved in, for example, protein trafficking, or like, deposition of the protein into the cell membrane, or things like this. To your first question, there's quite a lot of work in this space now. So for example, doing CRISPR testing of eQTLs, it's still kind of challenging because you need to do exact base pair editing. It's easier to do, it's easier to knock out we knock out protein coding regions, but it's also easier to knock out, for example, whole regulatory elements. And there's quite a few, there's several groups that are working in that kind of space. So there's a paper from Jay Shindori's lab, which Molly Gasparini is the first author on. And so she has this large experiment where she used CRISPR knockouts of a large number of regulatory elements to identify regulatory elements that are affecting expression. And then the other kind of technique that's really important in this space now, which I didn't get a chance to mention, is there is some methods called perturb-seq or crop-seq, where basically what they're doing is knocking out, like you knock out one gene and then measure RNA-seq expression, and then that's done in parallel with single-cell sequencing. So I think of that kind of experiment as being sort of logically the reverse of what we're doing. So in the perturb-seq approach, they're taking a gene and then looking at effects of downstream in the network, whereas we take a gene and then look at what are the upstream regulators for that. And so actually the follow-on work that we're doing to the screen that I showed you is actually, doing down, like we look for upstream regulators and then we do knockouts of those to look downstream. And so our idea is that kind of the coupling of the up and downstream version sort of allows to fill out networks. At least, that's what we're trying to do there.
Thank you. Very interesting.
Yeah.
Thanks.
Yeah.
Thanks, Jonathan. We've got another question here from Jobran Chebib. Jobran, if you can introduce yourself, please.
Hi there, Jonathan. Thank you for the talk.
Hi.
I'm Jobran. I'm at the University of Edinburgh. I'm interested in evolutionary genetics in general. I do a lot of theory work on pleiotropy, more specifically, and mutations. I wanted to just mention one thing that I think is really important, is that none of these traits that are being measured are working in isolation. And many of them have pleiotropic genes affecting not just those traits, but other traits. So you might be seeing a signal that is caused by selection on not the trait that you're measuring, but some other trait as well. So my intuition is that the genetic architecture that you'll be seeing will be like on the spectrum between polygenic to Mendelian trait, will be highly affected by how kind of modular those kind of pleiotropic effects are on that trait that you're measuring, and because it could be constrained by other traits or affected by other traits. So how are ways to get to ensure that what you're measuring is actually the genetic architecture of the trait you're interested in versus other traits that are driving the signals?
I'm not sure if I completely understand the question. So, I mean, I fully agree that there's has to be huge amounts of pleiotropy on many variants. Could you just rephrase the last part of the question again?
Sure. So you're interested in the genetic architecture of the trait, right? So you do these GWASs and then you get some kind of measurements of effect sizes of different sizes for the different gene variants. And you want to know, well, is that being driven by selection on that particular trait, or is it being driven by selection on other traits that you're not measuring? I'm wondering how you can distinguish those kind of things.
Are you referring to, okay, so I mean, a bunch of the talk's not about selection. You're referring specifically to the sort of the flattening part of the talk.
That's right, yeah.
The end?
Right.
Yeah. Okay. So for that, I completely agree. So the model that Yuval and Guy first developed, like they use this model that comes from I think it's like a Fisher geometric model where like you have this n-dimensional trait space, and then a mutation sort of you can think of that as being an angle in this n-dimensional space, and so then the axis that you're measuring is sort of a projection of that vector into one dimension. And so the effects in the in the context of the flattening, so in general the high pleiotropy has the effect of sort of increasing the selection on every, on all the variants. So it sort of shifts the point where that flattening occurs towards lower effect sizes. Because in a sense you don't only, like if you think of, like if we measure effect size on the trait that we're looking at, then selection is due not only to that effect size but also to the effect that it has on everything else. So it's sort of shifting the transition point. But what we don't have a good handle, so that part of the theory is sort of clear enough. But one question is whether, for example, like are the core genes where we get the biggest effects, are those likely to be more pleiotropic or less pleiotropic? And I think we don't really have a good handle on that. And I think it's going to be completely particular to the core genes and the traits. So I think the practical implication of that is that when we look at the top hits that come out of a GWAS, well, number one, we expect that the biggest hits are probably flattened down by selection, but the degree to which they're flattened may depend a lot on what kinds of genes they are. So, perhaps transcription factors are more pleiotropic than solute carrier genes. And so maybe then the transcription factors will be more, their signals would be more reduced relative to their biological importance than the solute carrier genes would be. So, I think this is it's a really important thing where I think we sort of understand it in theory, but actually how you translate that into data analysis is really challenging.
Yeah, thank you.
Thank you. Thank you.
Thanks, Jonathan, for taking also this question. And I think there are no more hands that I can see. So with this, I think, there is Antoine who would like to say something. Antoine, please go ahead. Please unmute your mic.
I'm coming back to the biometrical model. And one of the outcomes of your study is, as you mentioned, is genetic prediction, right? So now in the more biometrical approach, currently you may use genomic relatedness in a population and estimate genetic values for threats using thousands of markers and have a rather good estimate of the genetic values. This is actually used in genomic selection, or you can also use it in natural population. Now, how can you model, adding your model in this system, how can it increase precision of predictions? Can it upgrade, improve it, or do you see it, well, where do you see, where would it fit best?
Yeah, so I guess I think about the okay. So when I think about complex trait mapping, here I'm partly motivated by the work in humans. I think there's sort of two main use cases that people think about. One is, they think, one is the hope that you can predict risk, means, using polygenic scoring. And then the second is the hope that it gives you insight into the biology of a trait. And so I actually don't think that our model adds that much to the polygenic prediction part of this, because at least for an additive model, you don't really care what the, sort of what the mode of action is. I really think of it as being helpful for, sort of more of a conceptual kind of question, like, how do we understand the the mapping from genotype to phenotype, and which variants should we care about? I mean, one consequence I think of this is that, probably one shouldn't care about tracking down the causal variants for all genome-wide association hits, right? Like, some of them are going to lead you to really important genes, but if there's 10,000 genes that are associated with a trait, it can't possibly be that all of them are interesting to track down. So I think of it as being mainly on the mechanism side.
Okay, thank you.
Yeah.
Okay, so actually with this, we ended our series of questions, and we have also joined the end of this seminar. It was an honor to have you here, Jonathan. We are really happy that you could accept this invitation, and we're really grateful for that. So thank you once again for being here, and thank you for the audience for joining us here today online and engaging in the discussion. The recording of this webinar will be on our EVOLTREE YouTube channel. And also, if you want to comment or leave your comments, we are present on Twitter. I'm just pasting here in the chat the Twitter handle. So thank you again, and we will see you next week on Wednesday, same time. We have Emily Josephs from Michigan State University. So stay tuned with us and see you next week, same time. Thank you very much.
Thank you. Thank you, Jonathan, for being with us.
Yeah, thank you, Eva. Thank you, Santiago. Thanks, everybody. Thank you, Antoine. It's a great pleasure to talk with you all, and I look forward to hopefully meeting you all in the future when we can travel again.
Hopefully soon.
Yes.
Yeah.
Hopefully one day. All right. Thanks so much, everybody.
Thank you.
Thanks.